Good afternoon, everyone, and welcome. I'm Stefan Harrer, Director of the AI for Science program here at CSIRO, Australia's National Science Agency. Australia's Indigenous people are our first scientists and have the longest continuing culture on Earth. Their capacity to adapt and innovate in response to the challenges of the natural environment is something all Australians can learn from. So I would like to begin by acknowledging the Bunurong people as the traditional owners of the lands I'm joining from today, and I want to pay my respect to their elders, past, present, and emerging. Today marks a truly exciting moment for CSIRO, the official launch of the MVP of Sciansa, our agentic AI-powered lab assistant built by scientists for scientists. Sciansa has been designed to empower researchers, engineers, and innovators to think bigger, move faster, and to explore new scientific frontiers. It combines the latest advances in AI with rigorous scientific best practices to deliver safe, secure, and responsible assistance at every single step of scientific discovery. Over the past year, our teams, scientists, engineers, designers, have poured their expertise and creativity, their passion, into creating Sciansa. We've built prototypes, we've iterated through alpha and beta MVPs. We conducted countless design sessions, debugging cycles, and user tests. And now, we are thrilled to share with you the first release candidate of Sciansa and the vision driving it forward. So here's what you can expect in today's session. First, we'll introduce Sciansa and show you what it can do. You'll see it in action, from high-resolution genome annotation to brainstorming entirely new research ideas. You'll hear directly from early adopters and testers within CSIRO who have been shaping and using Sciansa in their scientific work. We'll share some of our plans for the external early adopter program, including how you can join the community of Sciansa users who can register and get on the shortlist for early access. And finally, we'll give you a glimpse of what's next, a look into the crystal ball, where Sciansa is heading as we scale up its functionality and as we grow our user community across Australia and beyond. And then, of course, we'll wrap it all up with a Q&A in the end, which is your chance to ask literally anything about Sciansa, the early adopter program, and the future we're building together. So let's dive in.
I'm Stefan Harrer. I'm the Director of AI for Science at CSIRO, Australia's National Science Agency. I lead a team that develops AI for scientists that helps them to be more productive, more creative, and accelerate the pace to scientific breakthroughs that would otherwise have been out of reach entirely or decades away. Sciansa is a trusted AI-powered tool that scientists can use to enhance and augment their scientific discovery workflows. From hypothesis generation and ideation, to designing experiments, to analyzing the outputs of these experiments, Sciansa will be by their side to help them perform these scientific discovery tasks at scales and possibly going into directions and tackling ideas that they otherwise would not have come up with.
The platform is incredibly accessible for users with no background in machine learning or artificial intelligence. This was a critical design choice for the platform. A lot of our user testers made it very clear that they didn't want to become AI engineers, but they wanted AI and ML tools that just worked and were validated. We have designed the product to make sure that the users incrementally develop trust in the product over time as they use it. The notion of calibrated trust is a crucial design principle for the product.
One of the biggest aspects of the scientific process is orthogonal thinking, which is thinking about 10 different concepts at once or 10 different aspects of a concept at once, bringing it all together and combining it, none of which AI models and AI platforms can do natively. Sciansa does that. That is the basic principle behind which science operates. The other thing that Sciansa does, because we are collaborating with scientists to develop solutions for scientists, it is completely and deeply rooted in the idiosyncrasies that a scientist needs and captures across their entire career. It both enables experts to enhance their expertise and make sure that it is available to others, and it enables non-experts to learn another expertise or learn another language of communication faster. So I believe that the impact Sciansa can have on science is to completely democratize the way knowledge is stored and knowledge is distributed in a scientific process.
Inside Sciansa run very bespoke, custom-developed models and tools that our scientists here in CSIRO have developed. We have a whole suite of robotics models and tools that we'll integrate into the platform as well. That is particularly exciting because it allows us for the first time to bridge the digital with the physical world, to help a scientist not only design an experiment, but actually get help with performing it in the real world as well.
Sciansa is a game changer for science because of how it brings agents and tools together to really accelerate the scientific process and bringing about new scientific discoveries, enabling the scientists to really hit those discoveries that we never imagined possible.
All right, so let's bring together what Sciansa actually is, the key parts, the key features of it. Sciansa is an agentic AI platform that is designed to infuse AI at every single step of the scientific discovery method. It empowers all scientists, regardless of whether they are AI wizards or completely newcomers to the field, regardless of their depth of understanding of AI, to collaborate with generative AI and significantly accelerate the path to scientific discoveries. Unlike other tools that might apply AI to niche, isolated tasks, Sciansa is able to integrate AI through the entire research process. It's truly broad AI. Our agent-based intelligence that manages tasks, offers recommendations, and understands the user's goals, provides a level of integrated support beyond typical automation. Our ultimate benefit here is to spark two things: creativity and productivity. We believe that by using Sciansa, scientists can accelerate the path to breakthroughs that might otherwise have been decades away or out of reach entirely. So let's look at some of the key features in the platform. First one up, and I think one of the most important ones, is Sciansa is a collaborative AI tool. It enables scientists and researchers to work with AI, not positions AI instead of scientists, instead of humans in the workflow. It's a collaborative, augmenting tool that scientists can use to scale up and augment what they are doing in their workflows. Next one, equally important. You will see that Sciansa has deep scientific domain-specific capabilities, but it also has completely domain-agnostic capabilities that span all scientific domains. We have built into Sciansa a set of responsible AI and guardrails. Right? That is very important, especially if we look into scientific best standards of practice. We need to make sure that there's data privacy, there's transparency, reproducibility, explainability, and reasoning through all of this, paired with human oversight. That's what you'll find in Sciansa. Sciansa is a workflow and task management tool, right? It creates workflows intelligently. A user can start by entering a prompt, but then the magic really starts. Our agents will generate the workflow steps, And the user can then verify, adjust, change, and manage all these interdependencies of tasks and information. And that can be shared between steps and topics, all highly integrated and accessible to human scientists. Finally, the platform is completely cloud adaptable. Whether you want to run this on-prem or in the cloud or in a hybrid version in between, Sciansa can do this. And it also does not require you to have any specific cloud provider infrastructure. You can run this on any cloud. So we believe that this probably satisfies any organization's requirements. Sciansa brings together data, tools, models, and agents into one unified ecosystem. It breaks down the silos between disparate resources, and it makes it easier for scientists to navigate and conceive complex workflows. Now, without further to say, after these rather theoretic musings, we think it's time to actually show you the platform and how it actually works in reality. So over to Rahul Rain, who is the owner of the agentic AI experience in our Sciansa team, and he'll demo some of the capabilities of the platform to you now.
Sciansa is designed to be a platform that works alongside a scientist. It collaborates with the scientists by making sure that they have access to AI agent teams to answer all the required questions. We start off with logging in, and the login is directed through Australian Access Federation, which allows any university and academic organization in Australia to connect with the platform. Once we click the login button, it will take you to a list of organizations that can be used, and we would select CSIRO in this case. And once you hit continue to your organization, it will log you in. Once the user is logged in, the front page essentially takes them to their last visited project. Inside the platform, the user can choose to create a project or resume an existing project. To create a new project, the user would simply click the Create a Project button and enter the name and description for the project. In this case, I'm going to call it Platform X, and I'm just going to add a dummy project description to it. Once the project is created, they can have the ability to create new workflows, and this is where the fun really begins. To start using the platform, the user would simply need to add their research question or topic into the chat box. In this instance, I want to look into Listeria monocytogenes, which is a pathogenic bacteria, and also try and identify what genes give it the ability to be pathogenic and study the mechanism at a protein level for these genes by focusing both on literature search as well as some actual bioinformatics. The user can then press send, and they will start seeing the platform chugging along, trying to both identify the steps that it needs to take to answer the question, as well as recruiting the appropriate agents. Once it has done that, it will come up with a particular workflow. In this instance, it has suggested that I do a unique gene finder workflow step using the unique gene finder tool to identify novel genes, followed by deep research, which is analogous to a literature search. Now, if I don't like this, or if I want to regenerate the workflow, I simply need to click the button and add into the box my comments. In this case, I wanted to click regenerate workflow button, and I asked it to please ensure that we do a comprehensive literature review before any analysis. I also wanted to look into the additional aspects of pathogenicity and regenerate the workflow. I also want to look into additional aspects of pathogenicity, and then I click the regenerate button. Here the platform will continue to go and recruit the right tools for the job to address the additional comments that you have provided. Once a new workflow has been regenerated, you can see that it has followed my instructions to include the literature review analysis before any other bioinformatic analyses, and it has added an additional disease mechanism investigation agent step, which will look into the mechanisms. The other thing that the platform does is to make sure that the prompts are appropriately formatted so that it suits the requirements of the tools. Also, so that the user does not need to do any kind of prompt engineering. They can simply work with that and move forward, or they can build upon it to improve the outcomes even more. To get started, we can simply start the workflow. Once you start the workflow, it takes you to a page where you have all the tasks set out as well as the original query. It also brings a canvas view which is designed to identify and visualize the relationship between all the different tasks. This is important as an aspect of the platform, as we wanted to maintain the ability to have orthogonal thought. Each task may not be related to every other task. Alternatively, in this case, we have 3 tasks, but we don't want to give the disease mechanism investigation agents simply the literature review. Instead, we also want to give it the list of genes that will come out of the analysis between Listeria monocytogenes and its non-pathogenic cousin as genes that may be responsible for pathogenic behavior. To get started, the user would simply click the Run button with the first step. The steps that have not finished a preceding step will actually show up with a warning and will not be actionable. Once you hit the Run button, it submits the job and starts the run. One of the salient features of the platform is that it is not limited to simply one domain or one species. In this case, a user wants to understand Bt transgenic cotton. They ask about its gene discovery, protein structure, and importantly, how to tackle emerging Bt toxin resistance and find new IP with freedom to operate. For a biosecurity challenge, a user may want to develop species-specific RNAi for, let's say, fall armyworm pest that doesn't affect similar species, and they can ask for gene targets, IP and regulatory landscape reviews, as well as brainstorming. Now that the first job is finished, you can see that the second job can now be executed, and we can now run this. As you can see, the prompt has already been pre-filled and is actually trying to find unique genes in Listeria monocytogenes that are absent in Listeria innocua, presumably as being candidate genes that may impart pathogenicity. I am happy with the prompt, and we can simply press Run, and we'll come back to have a look at the results. The analysis is complete, and you can see the outputs from the analysis much the same as you would see output from the agents. This can now be passed on into the next step, and that allows us to create an entire workflow. That is not all we can do. While we are doing and running the workflows, we can also chat with the entire platform and ask it to start curating any particular list. So for example, I can ask it to identify any toxin genes that came up in the analysis, and also ask it to help us with identifying the function of those genes in extreme detail. We can also ask it to suggest any tools and prompts that the platform has access to achieve the same outcome. As you can see, once the output is produced, it can also tell us the tools that we can use next. It will give us example prompts. And in this case, for example, it has suggested that we use the Gene Whisperer tool, which is one of the bespoke tools in the platform, and pick a gene and then help us identify the function for the same gene. The user can either select that prompt and add it into the next step, or they can choose to edit that prompt further when they add the step as well. To do this, the user would simply click on the Add Step button in the main workflow page and select the Gene Whisperer tool and add the prompt into the prompt box, thereby now instructing the platform to identify the function for the gene in question.
Wonderful. So now that you've seen Sciansa in action, let's take a closer look under the hood. The platform brings together a rich ecosystem of bespoke AI models and agents that fall broadly into 2 key categories. There are those specialized in health and life sciences, built for biologists tackling domain-specific challenges. You've just seen a deep dive into those. And then there are those that are completely science domain agnostic, designed to support all scientists, regardless of their field. right? So you might have seen a deep dive into genomics right now, but if you're an astrophysicist or if you're a socioeconomist, you can use the same principles here. To bring this to life, we have selected and we'll show you one example from each category. First, Sonali Mayumdar, genomics researcher and software engineer, will walk us through a best-in-class genomics model developed by CSIRO scientists and now available directly through the platform. And then you will hear from Rahul again, who will introduce us to an AI agent crew purpose-built for scientific brainstorming, for ideation, for hypothesis generation, a powerful tool for accelerating discovery across any domain. So, over to Gene Whisperer.
Thank you, Stefan, and welcome everyone. I'm very excited to introduce Gene Whisperer. This work was done in collaboration with scientists, researchers, and engineers at Ag and Food, Environment, and Data 61 RU. The main aim of Gene Whisperer is to be your bioinformatic assistant and to push the boundaries of our coexistence with an AI. It is a tool that leverages the power of large language models to enhance genome annotation. Today, I'll give you a brief overview of how we build the tool, demonstrate its capabilities, and how it helps supercharge genomic research. Gene Whisperer is an LLM-powered multi-agent system capable of performing deep research tasks such as gene trait association, gene function recommendation, targeted gene discovery, or even literature retrieval. Seamlessly integrated into bioinformatic workflows, Gene Whisperer enhances annotation efficiency, accuracy, and consistency. At its core, it orchestrates a set of sub-agents such as NCBI or Entree agent, UniProt, BLAST, Gene Ontology, and PubMed agents that collaborate towards an outcome. It is currently deployed, running, and available to you within the Sciansa platform on Google Cloud. Our bright researchers at Sciansa have been using Gene Whisperer extensively to support and further their research goals. Here are some shining examples. Our first use case is a gene trait association study. Imagine a research scientist has generated a list of candidate genes from prior analysis. They want to identify which of these genes are most likely associated with a specific trait of interest. All they need to do is to enter a prompt describing their query. In this example, the researcher is asking whether GAI, like protein 1, is associated with wheat height, and if so, how. Gene Whisperer aggregates information from multiple data sources and identifies the gene as producing the DELA protein RHT1. It highlights the gene's biological role in gibberellin or GA signaling pathway and notes that the mutations in this gene are linked to reduced plant height. It also generates a detailed report on this protein along with conclusions that are backed by verifiable literature references. In our second use case, we have chosen an instance where Gene Whisperer is very useful for scientists working on non-model species that don't have a well-annotated genome. The raw sequence of the genes can be given to Gene Whisperer to gain a more detailed description of the candidate gene that stand out in the result of genomic analysis. This makes it easy for researchers that are not gene annotation experts to interpret the results and to identify the functionality of the genes. That brings us to the end of the sneak peek into Gene Whisperer. To know more, please connect with the Sciansa team and check out the Sciansa platform. Thank you.
Two parts of any scientific process. There are the subjective components and the objective components, such as Gene Whisperer or other tools that have a definitive outcome or an prescribed outcome. For the more subjective components, where you can either generate hypotheses or you're looking to generate ideas, dig into inference, that's where the brainstorming copilot really comes in. It is designed to help users either dig into a particular idea or generate novel hypotheses based on existing referential material. It adapts to the user's requirements, thereby creating more relevant ideas, and acts as a more impactful copilot. It is also dynamic in nature and recommends more appropriate brainstorming strategies to the users based on the queries that they have actually asked the copilot. The brainstorming copilot has three modes. It has the breadth mode, where you can explore a wide variety of unrelated concepts. It has the depth mode, where you can actually dig into a very specific concept and generate much more focused hypotheses and ideas in one area. or the hybrid mode, which is the default mode, where the agent automatically decides the trade-off between more depth or breadth, so that it adheres to the user's requirements for any specific idea generation exercise. The way this works is very straightforward. It's when the user submits a particular query and they either give it earlier steps from the workflow or they give referential material to the copilot, it finds the most appropriate mode for the said query, and it sets the direction, the North Star of the ideation process. Once that's done, the agents then do a lot more focused research into each and every one of these areas and collects all the relevant background data for all the different concept areas that we would engage in, either with the breadth mode or the depth mode. Then the agents actually adapt the brainstorming process again to the user's requirements so that the outcomes are more relevant and guided by the user's intent. It also then triages the background data and the newly discovered data to fit that direction. And then finally, the agent scores each and every one of these ideas based on a set of practical criteria, if you will, so that it can provide the user more appropriate outcomes that can help actionable decisions. The net outcome of the entire process is to both help improve efficiency in the innovation process and align ideation to strategic intent. The ideas generated are steerable and hence more pertinent and fit for purpose. And finally, the practical and realistic scoring of every single idea ensures that the user has more information on hand to evaluate each novel or repeated idea or hypothesis before acting on it.
Cool. So we've just taken a look under the hood of Sciansa. I think it has become clear that this is not just a chatbot. Chatting is the thing that makes it so easy to interact with, but under the hood run models, agents, data sets, the tech that makes it all possible. So Sciansa runs this really for you. It doesn't just talk to you. At the end of the day, though, technology is only as powerful as the people who use it and who trust it. So that's why the next part is probably one of my favorite part in this webinar. You're going to hear about from some of the scientists who have been using Sciansa from the very start onwards. They've built models that are now part of the platform. They've been testing them early, and they are already using these models to push the boundaries of their very own research. First up will be colleague biologists Rob Spade and Dan Steinberg, who will share what motivated them to get involved and how Sciansa is transforming the way they and their teams work. And then we'll hear from roboticist Brendan Tidd, who will give us a glimpse into how Sciansa is opening up new frontiers in automated experimentation and lab organization. This is all where Sciansa really comes to life through the scientists who are shaping it and who are shaping the future with it. Over to you, Dan. Oh, Rob, sorry.
Hello, my name is Robert Spade. I'm the Director of the Advanced Engineering Biology Future Science Platform at CSIRO. We have around 20 projects looking at different transformational aspects around how we can engineer biology to solve big problems, as well as socioeconomic research and the engineering of biomolecules, organisms, and production processes. We have a big focus on leading-edge AI and machine learning tools that generate new biological insights and knowledge, as well as predictable designs for proteins, organisms, and fermentation processes. Large, high-quality data sets are essential for model performance, and we integrate biofoundry-level, high-throughput wet lab biology with dry lab model generation, training, validation, and function prediction. The collaboration with Sciansa is essential to get our tools into the hands of working bioscientists, making it easy for them to work at the cutting edge of AI biology. On a personal level, as the director of the Future Science Platform, I use Sciansa every day to explore research topics, novel hypotheses, the competitive and IP landscape, and help develop research strategy. I do this both for our existing projects and for the many new project opportunities with companies coming across my desk. Sciansa is already an essential tool for me, and I am very much looking forward to our new science tools and engineering biology workflows getting onto the platform and into the hands of research scientists. Thank you.
Hi, I'm Dan, and I'm a machine learning researcher in Data61. Today, I want to briefly present to you an example of how I think Sciansa and new generative AI models can change how we use machine learning in science. The example I want to present is a workflow we created that uses machine learning for engineering better enzymes that degrade plastic-like molecules, with the aim of using these enzymes to manage plastic waste. The enzyme we started with is known as PPS, or aryl esterase, and I've given it sequence and structure on the right. This enzyme reacts with esters, a compound of plastic, to break them down into acids and alcohols. And at CSIRO, we are actually lucky enough to have access to a biofoundry, a robotic experimental platform, where we can create 96 different enzymes and test them at once for how effective they are at breaking down these esters. So the workflow we have used to find these enzymes is on the left. It starts with our in-house protein engineer doing a literature search and using their expertise to select PPS as a promising starting candidate. Then we use a web-based sequence similarity search called BLAST to find 1,500 existing protein sequences that are similar to PPS. Then we test all of these enzymes for their ability to degrade esters using our biofoundry. Using the data from the biofoundry, we train a machine learning predictor to select 200 more promising sequences for further experimentation. And finally, we test these remaining 200 and find that they are generally more active than the original 1,500 sequences. Now, how could we reimagine this workflow using Sciansa and other generative AI models that we are building? Well, we could start by asking Sciansa for a batch of 96 promising enzymes to degrade esters, and it could then use its inbuilt knowledge or a literature search or other tools to do this. We would then measure these 96 enzymes with the biofoundry, and this data would then be used to inform a generative AI model for optimizing these 96 enzymes in a process we call active generation. Sciansa could then be used to visualize these new enzymes and the experimental progress before feeding these new enzymes into the biofoundry and beginning the process again. So I believe that using Sciansa and active generation in this way could lead to us finding better enzymes than the original workflow and more rapidly. Furthermore, what I have described is quite a general workflow that could be used with other proteins. Thanks for listening.
Thanks, Dan. My name is Brendan Tidd. I'm with the robotics team in Sciansa and a researcher in the Robotics Perception and Autonomy group of Cyber-Physical Systems. We're developing a physical interface to science, helping you to run real-world experiments and robotics with robotics and intelligent labs. Our first use case is digitizing the insects in the national collection. There's an estimated 13 million specimens, 95% without a digital copy, holding the secrets that are critical to climate modelling and rich for new discoveries. We've developed a multi-camera turntable system that targets high throughput and the extraction of the precious label data underneath the insects. We're reducing the delay of accessing this data from centuries to years. Our digitization system needs to feed, and humans only like to work for 8 hours a day. We're using robotics to supply the digitization device 24/7 with the potential to scale to parallel systems. What you're seeing here is a controller that's been trained by collecting expert demonstrations from teleoperation using the latest robot learning frameworks. This pipeline enables us to quickly adapt to new use cases. More generally, we are automating physical experiments where robots can be used to prepare samples, as you saw with our robotic manipulators, and to interact with dedicated pieces of scientific equipment, for example, the camera system you've seen to digitize insects or the biofoundry that Dan mentioned. Through Sciansa, we're designing a tool for building workflows that interface with our robots to enable scientists to run physical experiments in their own labs. These workflows can be created and modified by scientists using Sciansa agents. In the coming weeks, we'll have a dexterous mobile manipulation platform coming online to run real-world experiments in our own lab. Thanks, Stefan.
Wonderful. So you've seen a snapshot of what Sciansa's capabilities are at the moment. There's much more to come, right? So let's look a little bit into the future, into what happens next. What may you expect the platform to develop into? Well, first one up is that we'll support ever more complex workflows. The agents, the tools, the models that you see in the platform right now have very deliberately been chosen to support some of the very early scientific use cases of those scientists that we started to work with right out of the gate in pursuit of their scientific work. That is a community, an ecosystem that will hopefully grow massively in terms of the diversity of use cases, in terms of the number of scientists, in terms of the variety in what they all do. So we'll bring on Nextflow workflows onto the platform that workflows that scientists have been using, and we bring those into the platform, connect them with additional agents and tools, all in pursuit of real scientific problems by real scientists in real labs. As this capability landscape grows, so will the responsibility landscape have to. So we'll expand our guardrails. This will include agent assessment and evaluations, benchmarking, performance frameworks, that we'll integrate into the platform. The ability to generate agents on the fly when no agent exists for a specific user prompt. That's very important, right? To actually grow the platform in its governance framework as its functionality framework expands. We'll bring on, as you've just seen in Brendan's presentation, a whole suite of robotics models, right? These will be robotics capabilities, which we'll develop with our colleagues at the National Collections and Marine Infrastructure Research Unit, supporting the digitization of the National Insects Collection, but we'll use that as a stepping stone to bring in physical haptic robotics capabilities that can be used in an experimental autonomous lab environment to connect the digital world with the physical world. And one of my favorites, right, which will make this such a vibrant community, is that we'll bring on the developer ecosystem later this year. This developer ecosystem is essentially the complementary community to the users. It's the contributors. So if you have built an agent, if you have created a data set, if you have built an AI model or not an AI model, that's useful, right, to be a part of this concert of capabilities, we will make that available to be integrated into the platform and infused into that ecosystem of agents that Sciansa supports. So that'll grow us in both user community and contributor and developer community, which brings us essentially to how does one engage with this platform? How can you get access to the platform? Because at the end of the day, right, and I can't stress this enough, right? A platform like Sciansa stands and falls with the number of users that trust in it, that use it, the number of different scientific use cases, the number of domains that it covers, the interdisciplinary aspect of what scientists are doing with it. So we need that community buy-in very, very much for it to be actually successful and trusted. As such, we have begun to craft an early adopter program. As a matter of fact, that's where we're coming to you today. We are very close to actually opening the gates there. And so what what that means is that, for the last 6 months, pretty much, we've worked through all the bells and whistles of, the contractual, legal, ethical aspects of giving such a platform into the hands of external users. And what we do now is we collect expressions of interest. And what that means is that we want to be strategic about how to roll out this platform in terms of the use cases it supports, the test cases it is being used for, the stakeholders we connected to, right? We want to make sure that the platform in its current state, in this state of the early adopter program, is actually valuable, is useful for users in its current state. That is, that it actually can be integrated as a valuable asset into these projects. So therefore, there needs to be an assessment as to, is the envisioned use case suitable for what the platform can do right now, or is it perhaps something that happens later down the road? We can also learn from these expressions of interest about what features might be desirable that aren't yet in our platform to put those on the roadmap. But this is sort of a stratification of use cases that we'll go through with all interested early adopters to make sure that we're really just giving the platform to those of you who can actually valuably and reasonably use it right now. That cuts both ways. As part of this adopter program, we will then ask feedback from our users. That's very important for us, right? So this is an MVP. This is something that will grow. This is something that will improve. This is something that will expand. And in order to do this, as a consumer-centric design exercise, we would like to hear from you what has worked, what doesn't work. And most importantly, we would like to ideally get evidence proof of the value add that this platform adds to you. So if you write papers with it, if you do something that qualifies as measurable impact, we would like to really know about this, because this, at the end of the day, is of course what we do it all for. So we'll go through this stratification process after reviewing the EUI submissions that we'll receive. We'll then select and shortlist the most suitable candidates, and we'll then get in touch. We'll talk. We'll have kickoff meetings. We'll prepare the platform so it can be actually used in the environment that you are envisioning it to run in. We'll onboard your team, and we'll establish all the channels that we need to then be able to constructively work together on testing and using this platform. So that's what's going to happen going forward. And you saw a QR code there. You see it here again. I think you've all seen the email address that we have shared as well. Behind this QR code, you will see a very short form that gets you on the essential initial shortlist. That will ask you for very high level information as to why are you interested in Sciansa, what do we like to do with it. There's no right or wrong answer to this. This is really just for us to get an idea where the interest comes from, and then to go about getting back to each and all of you with feedback on your submission in a strategic way. At the end of the day, we hope that this is something that interests you and your teams. It could be that you see an immediate use case for trying this out as part of scientific discovery work that you're doing right now. It could be that you have built a model, a suite of models, created data sets that you would love to connect to other such models or data sets as you know your capabilities to solve a scientific problem. It could be that you're implementing your data and AI strategy in your organization, nationally, internationally, enterprise, academic. There is no limit to this. And you might see a seed here with this platform to get a first step going in implementing this data and AI infrastructure pathway in your organization. All of these scenarios are highly interesting, and we'd love to hear from you and your teams about your ideas and plans of what you think you could do with Sciansa and what we could learn from you as you embark on that journey. With that, we'll go into the Q&A sessions. We'd love to hear and answer any questions that you might have. I already saw that many questions have been submitted already. We'll try to get through as many of them as possible in this webinar. I can also, though, assure you that we have collected all these questions, and if we don't get to a specific question of yours in this Q&A section in the webinar, we will still answer every question and make those Q&A answers available to every one of you after the webinar. Okay, so with that, let's go to some of the questions. I start with one in no particular order. We have one from Ed Butler asking, signing on through the Australian Access Federation is great. Can any organization that uses AF simply log in and start using it? No other steps for access? Well, yes, that is what will happen the moment we have established that your proposed use case is a good fit with the platform capabilities, and we've essentially onboarded you. But given that that has happened, it is as easy as that, right? So it's just logging on through AF, and then you can hack away. What else have we here? Okay, we have another one. Nathaniel Butterworth asks, when you say it is platform agnostic, how does that work? Is everything connected by API key using a standard framework? Yes, but I'll let Rahul take that one.
Thanks for that, Stefan. So yes, in a manner of speaking, there is a standardized framework, which is API driven, but the standardized framework is what we have developed as our backend. So the backend we have right now is a bespoke federated data storage and analysis framework, and that sort of underpins the entire platform that allows us to also scale infinitely while making sure that all the data is secure without directly exposing the data to any possible providers or endpoints or that may not have the privacy sort of requirements that are needed for science and innovation. At the end of the day, the user gets an absolute control over and the provenance of what happens in every to the data in each and every step, and that's how we're trying to maintain a standardization in it. Back to you, Stefan.
Thank you, Rahul. Next question comes from Anon. It is the following one. Does Sciansa provide links, references to each model data set used? It's a fascinating and great question that I think matters very much here. The answer is absolutely yes. Any external information that is used in generating an output is tracked. Source and queries that led to this output are made available to the user. User can actually do deep forensics, and it's not hard to dig this up. It's very natural to go into your workflows. You can, as a first step, break down, for example, inside a specific individual step, which crews have actually been contributing, which AI agents have been contributing and working together to produce the output of that step. You can then go into each of these crews and look up how each agent in this crew was designed. The rules, it's all out there. It will tell you what criteria it used to decide whether it has met an endpoint around accuracy or not. You will be able to go into really the DNA of every agent, in every crew, in every task, and that is readily available to you. You can even ask I think Rahul has shown this in the demo, you can ask after a step is completed, you can interrogate the output and ask for this information, and it will produce that to you. So it's designed for maximum transparency and explainability. You will also have, well, you might not have captured this during the demo, but if you rewatch it, you will see it, and if you use it, you see it out as well, that before a prompt is generated, the tool will actually give you the rationale, the thinking behind why it broke down your initial prompt into that specific set of tasks, what its thinking was, and it allows you to interact with it, to question it, to change it, but you always have the full picture in terms of what has been used, why has it been used, and is that information accessible or not? Yes, it is right there. That is what's important for scientific best practices work, and that is what we have hardwired into Sciansa. Okay. Another question, and I don't have the name from who asks here, but I'll put it out there anyways. Question is, what data structures can be supported? Yeah, that's a good one because there's so much different type of data in scientific work. Rahul, you want to take this one as well?
Sure. Technically, the data structures that are supported are entirely dependent on the tool and the workflow that we have that anyone's using inside the platform. So if it is a computational workflow, then the data structures will be for that specific computational or bioinformatic workflow. If it is agents, then it will be usually text and text-based or multimodal output. So it is completely independent of the platform. The platform is fully agnostic in terms of what data sources and formats it can interact with. They're entirely driven by the tool that the user is using. Back to you, Stefan.
Thanks, Rahul. Thanks, Rahul. Another question. Is the data you upload and generate kept confidential within an organization? Very good question. Again, full-heartedly, yes. It goes even further. Data that is generated within an individual project is kept confidential within that project. So you could have, theoretically, in your organization, you run a purely academic open source project where you want to have as many collaborators and you just want to reach everyone out there, and you could create a project in Sciansa that does that. And then you can have a project where you do some contract work for, I don't know, for a pharmaceutical company around what have you, like a commercial with a commercial aspect. And you absolutely do not want this to be shared anywhere beyond that specific project and those of your researchers who work on this. And Sciansa allows you to do that. So data privacy and making sure that there is no cross-contamination between projects is absolutely imperative, and it's built into every step of the platform. Okay, we have a question from Moji. For deep research, does it use general internet search or directly get papers from scientific databases? I'll give this to Sonali.
Thanks, Stefan. For the deep research agent, yeah, we are using the Google search mostly because we need to know from where the information is understanding the sources of the information. So yes, it is, but it can still go out to get any external information because from the web. So the answer is yes, we are using Google search.
I'll just add to that as well, that depending on the organization's agreement with respective data providers, it is very easy to actually plug in scientific databases as well. As a default, that would not be enabled unless such an agreement was already present.
Yeah.
Which I think also answers another question around whether access to publications from organizational subscriptions can be integrated into Sciansa. Let's see, we might have time for 2 questions. What have we here? Okay. Here's one from Graham Heary. What are the general and legal T's and C's associated with using Sciansa? Does IP generated in Sciansa vest in the end user only, or will CSIRO require a license to be negotiated? Is it provided as fee for access type of offering or license? This is a very comprehensive question. Try to go through it in a structured way. So the T's and C's of engaging with Sciansa as part of the early adopter program will be very straightforward. We won't have any any assumptions as to claiming rights to IP that our users will have created with using the platform. But in later stages, we envision scenarios in which all of these pathways that Graham has laid out here could be arranged. If the tool or a version of the tool, if a user wants to license it from us for pursuing a very specific scientific research program, we could imagine that happening. If we could also very much happen, as a matter of fact, I'm a strong advocate for this and a supporter, that a version of this tool with selected capabilities will be open sourced, right? That this is available for everyone, and for testing, of course, within there will still be T's and C's. I spoke about guardrails. I spoke about safety. I spoke about the ethics of using this. This we won't let go, of course. But for the purpose of using it freely, we could imagine that a version of this tool might be open source. And then if you look at other versions of tool usage, where a user might want to use this tool as part of a commercial project that perhaps we do together with you, where there's researchers and Sciansa users from CSIRO together with an external partner. Of course, such a scenario will include negotiating the IP terms of joint IP, but as a default, right, that tool is not built to actually capture all the IP that anyone using it creates with it. We want to actually be the custodians of this tool, and we want to be the providers of this tool. And as such, we plan to cater to very different scenarios of engagement depending on what our clients seek to do with the tool. I hope that is not too vague, but that's where we are with this right now. So bottom line is the EAP right now will be very straightforward. You will be able to pursue your use case. That will be your IP. We just are interested in your feedback and how it all works. That's what we want to be able to use to integrate into the platform to make it better, to actually scale it up and to make it even more powerful. Okay. Maybe one more question to close with. Can we upload data? It comes from Gerardo. Can we upload data to provide more context? For example, a paper, a protocol, etc. That is a very good question. We are making that feature pretty much available as we go. It is not active right now, but I think the moment the EAP will actually start and the first early adopters and users will use it, we'll have that function available. And we should say that this will entail that you can connect it to your electronic lab notebooks and not just papers, but any sort of data sources you might deem useful for infusing into a certain task, a workflow that you want to do. Okay. With this, I'd like to close the webinar. Thank you all very much for your interest in Sciansa, for lending us an hour of your time to show a glimpse of it to you. Hopefully, what you saw excited you. If it makes you curious, that's just as good. If you take nothing away from this but one thing, then it should be the QR code, which you please go to and let us know about who you are and what you would like to do with Sciansa. Everything else will flow there. We'll make this recording available online afterwards, so you'll have all this information there as well, and we'll answer all the questions that we haven't answered so far separately as well. Thank you all very much. Thanks to the team, thanks to the presenters, and looking forward to many exciting, fruitful discussions and projects with you, working with Sciansa on your cutting-edge science. Thank you. Goodbye.